Hello, so today we have chapter 21. We're going to talk about special senses. So I'm on page 741. There's a lot of cool pictures, and there's a lot of, well, not a lot, but there's a couple of tables that will be very helpful. Sometimes just looking at structures in an image can be a little overwhelming, but then that little table is going to tell you what each structure kind of their function is, and it's going to help you understand the picture a little more. So I would suggest you look at those tables as well, and maybe there's some questions from there that can be on the test. All right, so let's do this. Special senses. All right. So we're going to talk about the senses. We're going to talk about smell, taste, vision, hearing, and balance. We're going to talk about smell first, olfaction, or the sense of smell. So you have what's called olfactory epithelium, or kind of like the tissue, sense of smell. It consists of three kinds or three types of cells. You have your olfactory sensory neurons, your supporting epithelial cells, and your basal epithelial cells. And then you also have what these olfactory glands or Bowman's glands, what they call. So here's a little picture. As you can see, kind of maybe the mainly the olfactory area is on the upper area, superior nasal concha or concha. It's kind of like on the upper part, right? So we get on that, and that's where we smell. Our body's able to detect certain molecules, able to differentiate between smells. That's where we have our olfactory tract, which is here on the top, a little like orangish, yellowish. Um, we're gonna have our olfactory bulb, and then our epithelium, or kind of like a layer right in there. Um, so here's our bulb on the right. Here we're gonna see our different axons and terminals that are ending there to receive information, right? We have our tissue here on the bottom, our epithelium, who receives those signals and kind of send them through the olfactory sensory neurons. Um, we have our dendrites, cilia, little basal epithelial cells that kind of help out. Olfactory nerve kind of coming through this area as well. Here we have this little opening, the cribriform foramen. Remember, foramen is like a little hole, so kind of through there is where we send the information to the brain. Okay. These little molecules are going to be like the smell molecules, so you're able to detect what kind of smell it is through here, send those signals through that mucus layer in our nose, send it through the sensory neurons up to the brain. Okay, so here's a kind of like a tissue slide. We have again at the bottom olfactory cilia, and then these cells supporting epithelial cells. Here's our little duct, our connective tissue, basal epithelial, the sensory neurons right here as well. Our dendrite, receiving that info and kind of sending him through there all the way to our brain. Again, here's a picture of this little guy. It's probably an old picture, huh? Olfactory tract, frontal lobe, olfactory tract, and the olfactory bulb with that little edge right here underneath. Okay, again, this is kind of the same picture that we saw. Okay, pretty much there's a little pathway. We're not gonna get in depth. That's a little more physiology, but just basically a little pathway. You got your nerves, you got your bulbs, you got your tract. It's gonna get through. They're the sensations that reach the cerebral cortex without synapting in the thalamus. They go straight up towards the orbital frontal cortex. So that's pretty much kind of going through the nerves, going through the bulb to the tract, and then to that cortex. So that's a little mini pathway that goes through structurally. Um, Again, I don't want to get into the physiology of it because it's going to be a little more intense, but that's for another class. Okay, so we have smell, now we have a sense of taste or gustation. So we are on 21.2, 744. Okay, we have sour, sweet, bitter, salty, and umami. So all other tastes are a combination of kind of those, and they all depend pretty much on the presence of the molecule. So, for example, if you have a little sour, we got our basic molecules, which is an OH. We can get H pluses and give some sweetness. Sorry, the sugar molecules are going to give it some sweetness. The H pluses will make it a little bitter. So depending on kind of the molecules that are there, or the ions that are there, is what's gonna make it, what's going to affect it, and how what we're gonna taste and how we're gonna taste it. Okay. I think I got it mixed around now that I think about it. So we got sour is hydrogen plus ions. Okay. Salty is sodium ions. So that's why NaCl is our salt. So sodium ions is salty. Sour, is our hydrogen ions. Um, Bitter taste is when we get like caffeine, morphine, quinine, and also many poisonous things have bitter taste. That's why it's like a natural kind of instinct when you get something bitter to like spit it out, because a lot of those chemicals that are in the bitter foods are also in some poisons. So our body has a natural repellent against that. Um, Umami taste, first reported by Japanese scientists, is meaty or savory. So mainly amino acids present in food. Glutamate, monosodium glutamate. So yeah, so we got different types of tastes. Okay, then we have the receptors for taste. We got these taste buds. The number of taste buds decline through age, and each one of those has three types of cells. We got supporting epithelial, gustatory, and basal epithelial cells. So we got these little areas of taste buds. As you age, you're going to be losing those taste buds. So people that age more will kind of taste worse, or be able to detect the taste less. That's what it would be. Okay. And then you have supporting cells that have these little microvilli and these gustatory or tasting epithelial cells. We have what are called gustatory microvilli. So microvilli is kind of like little finger-like projections, and those basal epithelial cells. So here's our tongue, and we have this valate papilla, which is kind of like a V shape, kind of in the back. We have the fungiform papilla and the filiform papilla, and the foliate papilla here on the sides. So pretty much you're gonna have this little taste buds, right? So here's our filiform, fungiform. Here's our little taste buds that are gonna be in those areas, and inside that taste bud, we got these cells, and we got these neurons that are able to send the information of whatever we taste. Here's our little microvilli, gustatory microvilli, little finger likes. We have a test spore that kind of goes through, and then our cells will detect it and send it to our first order taste neurons. Again, a couple images. Here's our filiform, fungiform, supporting epithelial cells, basal epithelial cells. So here's our little pathway. We got our little tongue, right? Sensory neurons and information, our facial nerve, glossopharyngeal nerve, or vagus nerve, depending on the area. We'll send through the medulla, the gustatory nucleus, then to the gustatory cortex and insula of the cerebrum. So this little area. If you remember this like elongated face, kind of like look a little weird, right on that brain picture that we had last week. Okay, those taste buds are found in elevations, so kind of like little raised up, and you can kind of feel like little cells, like little elevations throughout our tongue. So those are the valate papillae, the fungiform, and the foliate papillae. So those are the three we saw in the picture. And the whole surface of the tongue has filiform papillae, and those do not contain taste buds, but those are little kind of cells that are also around there. So the pathway of that one as well, so not just how we saw the smell pathway, the olfactory, we got the gustatory pathway, the taste. So we got the three cranial nerves, as you saw in the picture as well, our facial through the left, glossopharyngeal more in the middle, and our vagus nerve. Those impulses will propagate along these nerves to the gustatory nucleus in that medulla oblongata and come up to the cerebrum and that cortex area where we're going to have the taste. Okay. So we got smell, we got taste, and now let's get into vision. Okay. Vision, seeing something. So in vision, we have our eyelids, our palpebrae, levator palpebrae superioris muscle, right? Levator is gonna like lift it up. palpebral fissure, lateral commissure, medial commissure, and our lacrimal caruncle. So those are kind of the structures of around the eyelids. I was reading that because I get it a lot. My eye, like I'm just like this, and then my eye starts twitching a little bit, and you got these muscles kind of like twitching a bit, and it's been being a little more lately. I'm like, man, what's going on? I probably have an issue, but no, it's just that I have a lot of stress and fatigue. So I was like, yeah, that makes sense. I got too many jobs. I'm a little too tired. I don't sleep a lot, so like my eyes are like super tired, and like they'll just start like one eye. It's mainly my left eye, and it'll just start kind of vibrating. My little muscles just, but it's mainly fatigue or stress. So yeah, just thought I'd share that out. Okay. We also have the tarsal plate, tarsal glands, calazione, the conjunctiva, the palpebral conjunctiva, and the bulbar conjunctiva. And those are kind of all the structures. So here's a little picture. There you're gonna see the bulbar conjunctiva, palpebral conjunctiva, kind of the different layers throughout the eye. Here's our levator muscle. Here's our superior rectus muscle. So our levator, see how it's attached kind of to like the upper eyelid, kind of in this area, so it's able to elevate it, right? Inferior rectus muscle on the bottom. So the eyelid on top has a lot more movement than the eyelid on the bottom. Okay. Eyelid, our eyelashes, our lower eyelid, tarsal glands. Here's our pupil, our lens, and our iris. Okay. Now we got our lacrimal gland, or how we get tears. Lacrimal gland, excretory lacrimal duct. So lacrimal gland is kind of secreted there, and then it goes through that excretory to kind of get it out. Then it goes, here's a little a pathway of those tears that we get to the surface of the eyeball, to the superior or inferior lacrimal canaliculi, which will drain it into the lacrimal sac, which is more in the middle, which will drain it into the nasolacrimal duct, which will then drain it into our nasal cavity. That's why you're all crying, and you got all these boogers coming out as well. So we got people just producing a lot. So yeah, little nasal cavity and all that stuff. The lacrimal apparatus produces and drains the fluid and the tears. So we just talked about that, the lacrimal duct, puncta, the canaliculi leading to the lacrimal sac, and the nasal lacrimal duct, kind of more inside our nose, almost. Okay. Extraocular muscles, or muscles kind of like outside or around the eye. We have a periorbital fat around it. We have 6 extraocular muscles: our superior rectus, inferior rectus, lateral rectus, medial rectus, superior oblique, and inferior oblique muscles. So also a little bit of the anatomy of that eyeball itself. So now we talked about the muscles around it. So the eyeball itself, it has a fibrous layer, vascular layer, an inner layer, and then it has a lens. Okay, and the book explains a little more each one, a little more in depth. Fibrous or fibrous layer, the outer layer of the eyeball. So I get a lot of allergies, and then this little section right here, I get like itchiness, and I start scratching, and then I start crying, and then I'm going through what I'm teaching, and anyways. So yeah, it talks about the fibrous layer, vascular layer, the inner layer. Here's a picture as well. Here's our muscles: lateral rectus muscle, medial rectus muscle, ciliary muscles over here. Light coming in, visual axis, kind of the area where we can see. The chambers: anterior chamber, posterior chamber. The cornea, the pupil, the iris, down here at the edge. And our lens. Here's our bulbar conjunctiva, kind of like at the edge almost. What else do we have? Our retina, here's one of the layers. Choroid sclera. What else do we have? So here's our visual axis. So that's why our eyes, have you noticed? Oh, I don't know. Well, you probably have. But have you noticed that when you take pictures, like people have red eyes, you're like, oh my gosh. I remember like a kid, I was like, oh my god, that person has a devil in them. Like you have like red eye, or like the picture comes out with people with red eyes, and you're like, no, let's take the picture again. And that's because the lights are focused on the blood vessels that we have on our eyes. So that's why they look red. But then when you just look at our eyes, they look dark because you're looking at the back, kind of like the back layer through the eyeball, and that's what makes it look a little darker. So the picture kind of light rays focus on those vessels, and that's why a lot of eyes pop up red in pictures, which is a little scary. But and now I know it's scientific. Now I know the science behind it, so I'm not scared anymore. So here's our little iris kind of coming on around the pupil. That's what I'm talking about. It's all dark. We're focusing kind of on the back part of that. That's why it looks dark. The ciliary process. This is a pretty cool picture. I'm not gonna lie. Pretty cool picture. Okay. So talking about our retina, so it has an optic disc, and there's a central retinal artery and retinal vein, right, to get some blood and to take blood out. We have a pigmented layer. That melanin in the pigmented layer will help absorb those light rays. We have a neural layer or a sensory layer, and then the three layers of retinal neurons: photoreceptor, bipolar cell layer, and the ganglion cell layer. Okay. The cells present in the bipolar cell layer, we have some horizontal cells or some amacrine cells. I don't know if let's see if it explains a little more in here. We have rods, we have cones, photopigment molecules, we have the macula, the fovea centralis. So we have here our different layers. So here's a picture. Okay, so when our pupil constricts, we have the sphincter pupillae muscles contracting. And then when our pupil dilates, we have the dilator pupillae muscles contracting. So depending on the muscle that contracts is if it's going to constrict or dilate. Right, and here's a little pupil in the middle. Okay, so a normal retina seen through the ophthalmoscope. See how it's all red now? Optic disc, the retina, blood vessels, fovea centralis, and our little macula in the middle. Okay. So more microscopic pictures of that retina. So we have here our vein and an artery, central retinal vein, central retinal artery. The optic disc here, like in the edge, and we have here kind of like on the layer around it, the layer of the retina, pigmented layer of that retina. That's why we have different colored eyes, right? Because we're looking at the back or the pigmented layer of that retina. Here's our rods, our cones. Here's our different cells that we talked about: horizontal, bipolar, amacrine cells in that bipolar cell layer. Because of those bipolar cells, inner synaptic layer. So we got some synapses going on there, sending some signals through. Ganglion cell layer, optic nerve and the axons, and the blood vessels coming in. The direction of the nerve impulses through the retina, direction of incoming light. So from the bottom to the back. Okay. Again, here's another cool picture. Those rods and those cones. They're pretty cool, huh? They all look like that. So simple, but yet so complex. Pigmented layer. So here's our pigmented layer. Photoreceptor. I always tell them I have colored eyes. I mean, brown's a color, black's a color, so I got colored eyes. Photoreceptor cell layer, bipolar cell layer, ganglion cell layer. So each one of those cells, we have a specific function. That's why you got different layers around those cells. Okay, the interior of that eyeball, we have the anterior segment, the aqueous humor, the anterior and posterior chambers, posterior segment, which I think there's a picture of that too. Right, if I'm not mistaken. Yeah, so we'll see a picture right now. The vitreous body, the vitreal floaters, hyaloid canal, and the intraocular pressure. So here it is. Our anterior segment, which is anterior chamber, posterior chamber. So here's our lens, our little iris. Kind of send the signals through anterior ciliary vein. It's coming out, right? Ciliary muscle, ciliary process, ciliary body, this area right here. vitreous body over here, the cornea kind of in the edge on the top. Okay. So just how we have the olfactory pathway and we have the gustatory pathway, we have the visual pathway. We got these bipolar cells, signals to the optic nerve, the optic chiasma, it's kind of where they cross, right? The nerve kind of crosses. The optic tract, lateral geniculate nucleus, and then it goes to our primary visual cortex, superior colliculi, and pretectal nuclei. So it's like a little pathway it goes through. So here we can see it as well. I think there's another picture. Yeah. So here's our optic nerve on the top. Here's our little chiasma, the crossing of it. Optic tract, lateral geniculate nucleus of that thalamus that we have, the optic radiation, and the primary visual cortex. In the back, if you remember, in our occipital lobe of the cerebrum, in the back region. Here's another cool picture of that little chiasma, optic chiasma, kind of crossing through right eyeball, left eyeball, to kind of cross over some uncrossed axons and some crossed axons. Pretectal nucleus, superior colliculi, lateral geniculate nucleus. I'm always tired. I feel like this time difference is not helping me out. Okay, so we got smell, we got taste, we got vision, and now we're gonna get into our ear, sorry, into our ear, and then we're gonna get into balance. Okay, so the ear, we got three main regions: the external ear, the middle ear, and the internal ear. So we got all those three. The external ear mainly has the auricle, the external acoustic meatus, the ceruminous glands, and it also has a tympanic membrane or the eardrum. Okay, that's in the external ear, which has the umbo and the malleus. So as you can see, so here's our auricle. So you'll see like the outside, the middle, and the inside. Okay. So we have here's also on the left a little legend of kind of like yellow, blue is the middle, and then kind of pink is like the inner on the inside. So kind of this area will be our middle ear. So we're on the outside right now. The auricle, basically our ear, the helix part, the lobule on the bottom. Can we get our piercings? Right, elastic cartilage, some ear wax in this area. Here's our tympanic membrane that's going to detect different waves coming through. The external acoustic meatus, the malleus, the little edge right here, incus on the inside. So now we get to the middle ear a little bit. Here's our temporal bone as well coming down. Our cochlea. So now we're in the inner ear, internal ear for sure. Once you're over here in our cochlea, semicircular canal, interacoustic meatus, vestibulocochlear nerve receiving those signals of whatever noise we got through. So it's like this little pathway how it travels through. It's pretty cool. And this little fella, the stapes in the vestibular window kind of vibrate in here. So receive whatever pressure it is through the ear, and you see a little vibration through there. It's pretty cool. Okay, that was the external. I think I talked about all of them, but we'll just get a little more. The middle ear is kind of small, air-filled cavity. Temporal bone that is lined by that epithelium. And in the auditory ossicles, you have that malleus, the incus, and the stapes. So it's pretty much, so let's see here, go back, it's right here. So here's our little malleus, the little circle, the incus, and then the little stape. It's down here. It's kind of like a little bell. I think there's another picture. Yeah, okay. The head of the malleus articulates with the incus, so they connect, and then the incus articulates with the stapes, and the footplate of the stapes fits into the vestibular window, or that oval window. You so have tensor tympani muscles, stapedius muscles, and then that auditory tube, kind of known as the Eustachian tube. That's horrible. Okay, so here's our middle ear now. So we get rid of the external now. So here's our middle ear now. Here's our malleus, our incus. Here's our ligaments, superior ligament, posterior ligament, lateral ligaments, now holding them together. Stapedius muscle right here. There's a little muscle in there. Tensor tympani muscle, you can see coming across as well through the tendon. And here's our little stapes that fits into this little canal that we have here. It's our little cochlear window as well. So kind of sending vibrations throughout and letting our inner ear, or letting our brain know what's going on. Okay. Here's more pictures. It looks a little weird, huh? It looks like so complex. It looks a little weird, but it's pretty cool. A superior ligament, the malleus. So here's our malleus. You can see, right? Tympanic muscle. So here's our muscle coming out, tendon coming out. Tympanic membrane, as you're gonna see. Here's our little stapes. Oh, look at that picture. That's so cool right here. Our little stapes coming across. Malleus and incus as well. Okay. And the third part, or the inner part, is the internal ear. It's known as a labyrinth. There's pretty much two main divisions. We have your outer bony labyrinth encloses and your inner membranous labyrinth. So perilymph and endolymph. Remember, peri is kind of like surrounding or outside, endolymph is kind of inside. Within the membranous labyrinth, it has those receptors for equilibrium and hearing, so we get a good balance and we can hear things. The three main areas on that bony labyrinth, we have semicircular canals, vestibulae, and the cochlea. So probably those are the words that you've heard about the most. So here's our external acoustic medius, the mastoid process, styloid process. So here's the semicircular canals and cochlea, kind of like on the inside, kind of going through. Kind of like a see-through, if you were like Superman, just looking through the bones. And here it is, the cochlea and the cochlear duct that we have here. Cochlear window down here. We have our little stape. Ampulla of semicircular duct. The utricle, the vestibuli, the saccule, anterior, posterior, and lateral are semicircular canals. So here's our membranous labyrinth, which has the inside lymph, and our bony labyrinth, which has the outer, or the epi lymph. Okay. Again, we got a couple more pictures. Here's our little incus, our malleus kind of sticks out, and our stapes. It's pretty cool, right? So that's our middle, and then we get into the inner, which is the auditory ossicle compared to the inner ear, compared to a dime. So it's just kind of giving you a comparison of the size, pretty much, and how big it is. That's why it's like so. It hurts so much when we got these big old sounds. You got neighbors putting music on all day, and like, anyways, vestibuli, cochlea. What do we have? Semicircular canals, anterior, posterior, lateral, kind of going through on the inner ear. The vestibule has the utricle and the saccule, and the semicircular canals have the ampulla and semicircular duct. Let me see if find that table that I was telling you about. I think there's more pictures. Yeah, there's more pictures. Let me see if I can find that. No. Okay. We have our cochlea, so kind of that curve that we have, that little spiral that looks like a what's it called? Un caracol, like a snail. Sorry, thinking in Spanish. Looks like a snail. It looks like Turbo. Huh. Anybody seen Turbo? You got a little Turbo over here, kind of running around. Okay. It's divided into two bony channels: the scala vestibuli, scala tympani. We have the cochlear duct and the helicotrema. Okay. Then we have our spiral organ, or the organ of Corti. And then we're going to see some pictures of that as well, which has the vestibular membrane, the basilar membrane, sterocilia and canucilium, spiral ganglion, tictorial membrane. Okay, so kind of looking through, remember, we're receiving the signals kind of from this side over here, from the outside to the middle. Here's our little stape. Receive those vibrations through the in. Those vibrations will kind of travel through and go around the cochlea, right? Scala vestibuli, scala tympani, kind of coming around. transmission of the sound waves from the vestibuli to the tympani by way of the helicotrema. So it's a little hole in the middle, helicotrema, and that's where you'll get that transfer of those sound waves between the vestibuli and the tympani. So where's our tympani? Okay, so coming in from the top, coming out from the bottom. Cochlear duct, cochlear window. There's a little more in depth, so kind of chop it up, that little area of the cochlea. Here's our vestibular ganglia. Here's our vestibulocochlear nerve. Why? Because it has a vestibular branch and it has a cochlear branch. So it's a nerve that combines the two. The scala vestibuli here on top, vestibular membrane, the cochlear duct, tectorial membrane, the spiral organ down there, the basilar membrane, scala tympani here on the bottom as well. So here's a section through one turn of the cochlea. So kind of through those little gaps, you have those waves coming out. And here's our nerve coming down. Cochlear branch. This is the cochlear branch of the vestibulocochlear nerve. Okay, so we hear through the different sound waves, so they're alternating high pressure regions traveling the same direction through some medium, and that's kind of what causes our hearing, or we're able to hear, depending on the sound waves. And they kind of give you nine steps, and you see those on the book as well. So it's kind of following the pathway through. Okay, so we have in our external acoustic medius, so we got this sound wave coming in, right? They strike the tympanic membrane coming through, the malleus, the incus, or stapes coming through as well. Then we had these stapes are kind of like moving, vibrating back and forth, right? Through the oval window, vestibular window, right, through 4, 5, and 6. So the movement of those, it's these fluid pressure waves. The stape plunges inward into the vestibular window. It pushes the perilymph kind of the scala vestibuli kind of through. Then those pressure waves are transmitted from that scala vestibuli to the vestibular membrane and into the endolymph in that cochlear duct. I'm following this chart or kind of mechanism of hearing on page 766. So if you'd like to follow it too, and it's also explained right here: the cochlear nuclei, lemniscus, inferior colliculus, superior alveolar nucleus, medial genucleate, primary auditory cortex, and auditory association area to kind of discern what's going on. Okay, so I was on 8. Pressure waves and endolymph. Remember, endolymph on the inside causes the basilar membrane to vibrate, move those hair cells of that spiral organ against the tectorial membrane. So there's a little spiral organ, move those little hair cells there. Tectorial membrane right here on the top. Here's our vestibular membrane. Cochlear conduct contains endolymph. Then the sound waves of those different frequencies will cause certain regions to vibrate obviously more intensely or less intensely, and then other regions. So then they'll be tuned for a particular pitch, which is pretty cool. That's pretty cool. It'll be tuned for a particular pitch. So since it's so narrow and stiff, you have high frequency sounds. We got maximum vibrations. And then the basilar membrane is kind of more wider and more flexible. And low frequency sounds cause maximal vibration of the basilar membrane there. So low frequency, and high frequency, and then high intensity. And we have the basilar membrane as well coming in. And then obviously the louder the sound is, the more stimulation of those hair cells that are going to move and be stimulated because of the sound. Sorry. but yeah. Okay. So then kind of following that auditory pathway. So that was just following a sound. Now following how it goes from that sound into our brain. We have the cochlear nuclei, lateral lemniscus, the inferior colliculus, superior olivary nucleus, the medial geniculate nucleus, the primary auditory cortex, and the auditory association area. As you can see here, here's our primary auditory cortex. Here's a little inferior colliculus, lateral lemniscus on the sides, superior olivary nucleus in the pons. Right, here's our cochlear nuclei in the medulla oblongata. Here's our little cochlear branch of the vestibulocochlear nerve coming in. Okay, receptors in the maculae of the ear. Then we have these little receptors kind of. So what's basically that picture what's saying is like our body's able to kind of adjust to that positioning over our head and kind of notice, keeping us balanced of how things are working and why things are working. It's kind of like a mechanism of equilibrium. So as we're moving to one way, the kind of gravity obviously moves them, so we're trying to adjust against gravity as well. So we have this little otolithic membrane and the little hair cells that are going to be here. Okay, so those receptors to keep a balance are stimulated by the linear acceleration or deceleration by tilting the head forward or backwards, and rotational acceleration or deceleration. That's why we can get dizzy, because we get a mixture of all this and our body's not able to recognize when we have certain acceleration, what we're hearing, what we're seeing, and what we're experiencing feels different to our body. The vestibular apparatus will have the saccio-utricle and the semicircular ducts. Okay. Then we have what's called these otolithic organs. Okay, that will contain the macula, the hair cells, supporting cells, the hair bundle, stereocilia, kinocilium, otolithic membranes, and the otoliths. And I think there's a picture here. So saccule and utricle, and then we have the semicircular ducts. So those are three: the ampulla, the crista, and the cupula. Okay. So here's our picture. Here's a little hair bundle. So here's our ampulla, crista, and cupula that you're going to see. Here's an ampullary nerve. You can see kind of the area in our inner ear. It's our semicircular duct. So that's gonna help us like be able to keep our balance. It's gonna try to understand our movement and set signals. So as the head rotates in one way, the cupula is dragged through the endolymph and bent the other way, which makes sense, right? It's like moving kind of like the opposite of what we're moving, trying to keep that balance. I got dizzy just by doing it. Okay, and then the equilibrium pathway. So just how we saw the olfactory, the gustatory, just how we saw. So we have smell, taste, our ear. Let me see. Vision, right? Vision pathway as well. Here's our equilibrium pathway. So it goes with the vestibular ganglia to the vestibular branch that goes to the VC nerve or vestibulocochlear nerve. It has our vestibular nuclei and then the inferior cerebellar peduncles. Okay, and the vestibular nuclei integrate information, vestibular, visual, and proprioceptors, and send those to the nuclei of cranial nerves 3, 4, and 6, to the nuclei of cranial nerve 11, to the vestibulospinal tract, and to the ventral posterior nucleus of the thalamus. Again, if you look, there's table 21.2 that kind of explains the different areas and kind of their function. The auricle kind of to collect sounds, the external acoustic meatus, it will direct those sound waves, tympanic membrane, kind of has those different areas. So it's a cool little picture. The inner ear, the middle ear, and the outer ear. So if you're able to if you want to differentiate between the structures of each one, that table will help you out. 21.2. I suggest you take an eye on that, so you can kind of see kind of what their main focus is. So here is a pathway, an equilibrium pathway, right? So we got this movement, it's going through, kind of tilting our head, anything like that. Vestibular ganglion, vestibular branch of the VC nerve, coming to the vestibular nuclei, to the different nerves, so 5, 6, 4, 5, the oculomotor as well, trochlear nerve, abducens nerve, accessory nerve, coming through to the vestibular area of the parietal lobe of the cerebrum, kind of the side area. All right, so that's it for that one. Again, I suggest go back and look at some of those tables, look at some pictures, try to understand those again, and I'll see you in class.